Okay, this is AP Biology review of Big Idea 2. Now at home, I highly recommend that you take out a piece of paper and follow along as I write these notes. Um, I go rather quickly, but I think it's really helpful if you do copy these and then listen to my explanations as you do that. And then you have these all available for you to review for the big exam. Okay, so Big Idea 2 states that biological systems utilize free energy and molecular building blocks to grow and reproduce and maintain dynamic homeostasis. Dynamic homeostasis. So that's a really packed sentence, and I'm going to break it down for us. Starting with the bit about biological systems utilizing free energy. So let's start with that. So we had a whole unit on energy. So I'm going to summarize it in a small picture. And we're going to be focusing on the two main processes that deal with energy, mainly photosynthesis and cellular respiration. Okay, so I'm going to use PS for photosynthesis and CR for cellular respiration. And the energy kind of cycles because the reactants of one become the products of the other. So the ultimate source of energy for our planet is the sun. It allows photosystems inside chloroplasts to harness that energy and to make glucose and oxygen, which then become the reactants of cellular respiration to provide ATP, CO2, and H2O are the products. And again, those become the reactants of photosynthesis. Okay, briefly, the two main processes of photosynthesis are the light reaction, which contains an electron transport chain in the thylakoid membrane. And the second major step is the Calvin cycle. Remember, that's where you fix CO2 into organic compounds. So carbon fixation in the Calvin cycle. In cellular respiration, we have the first step called glycolysis. This is a very ancient process. It's universal, found in every cell. If there's oxygen available, you move on to the Krebs cycle. This is where you harness high energy electrons and place them on coenzymes. They take those to the electron transport chain in the inner membrane of the mitochondria. Again, this is when oxygen is available to serve as a final electron acceptor. In prokaryotes, all of the electron transport chain occurs across the plasma membrane since they don't have mitochondria. Okay, in the absence of oxygen, cells can carry out fermentation. This is where they just go through glycolysis to make ATP. And the byproducts are either lactic acid or ethanol. Okay, so I think that's it for energy. Let's move on to this bit about molecular building blocks. So this goes back to our biochemistry unit, or the very first unit. So remember, organic compounds are made up of monomers. Remember, organic compounds are those that make up living things. And we can break those all up into individual units called monomers. And in order to make larger polymers, larger organic compounds, we have to join those monomers into a covalent bond. So the joining of monomers is called polymerization. And the way this is done is through the removal of a hydroxide group and a hydrogen group to form water. We call this dehydration synthesis. Dehydration synthesis. Okay, and this is how we form all organic compounds. And just a quick review, there are 4 groups of organic compounds. You've got amino acids, which form your proteins. We have, let's monosaccharides that form your carbs. And then we have nucleotides, which form nucleic acids like DNA and RNA. And finally, we have fatty acids, which form your fats or lipids. Okay, so that's your biochemistry review of molecular building blocks. All right, let's move on to this big vocabulary term, homeostasis. How do we maintain homeostasis? So that's going to be a big topic. So that's going to occupy the rest of this page. Let's first define homeostasis. Okay, so you should all remember that homeostasis is a process that maintains constant internal conditions. So even though the external environment changes, it's really important to maintain constant internal conditions for either the cell or the organism, because a lot of chemical processes depend on maintaining those constant conditions. Okay, on the left, I'm going to look at a few examples that we covered in class on how we regulate homeostasis. So how do we maintain balance? And this could be at many different levels, but some of the key examples we've gone over. Remember, inside prokaryotes, you have a collection of genes called operons, and they're regulated by an operator. So there's regulatory proteins that can either bind to the operator, activate it, or repress it. So go over that in further detail on your own. We talked a lot about enzymes and how important they are in cellular processes. So really important to maintain homeostasis and not make sure there's not too much of a product. So allosteric inhibition is one way to regulate how much product is formed. And lots of homeostasis examples in our endocrine unit on hormones. Insulin and glucagon, for example, work in opposition to maintain constant blood glucose levels. Insulin brings it down, glucagon brings it up. Okay, the key mechanism for maintaining homeostasis, remember, is called negative feedback. Negative feedback. Negative feedback. Okay, so I'm going to give you a specific example. It's not so important that you remember the names of these hormones. It's more important that you understand the process. Okay, so I'm going to go over one specific example using thyroid hormones. So thyroid hormones are produced by the thyroid, but they're actually regulated by two other glands, the hypothalamus and the anterior pituitary. Again, don't worry about where they're made, and don't even worry about the names. Thyroid releasing hormone produced by the hypothalamus goes through the bloodstream and affects secretion of thyroid stimulating hormones in the pituitary, and then that stimulates the thyroid to produce hormones. And then the thyroid hormones, once they reach a certain threshold, will stop the production of TRH and TSH. We call that negative feedback, to maintain constant levels of thyroid hormone. Okay, positive feedback has the opposite effect. Instead of dulling the effect, it amplifies the effect. And our most popular example is that of oxytocin, a hormone produced by the hypothalamus and then secreted by the posterior pituitary. And it actually is stimulated with the stretching of the uterus, causing the posterior pituitary to continue to produce increased levels of oxytocin, leading to increased uterine contractions during labor. Okay, moving on. Let's look at homeostasis at different levels, starting with the molecular level. Okay, when we're looking at the molecular level, it's all about enzymes. Okay, So enzymes, remember, need optimal conditions. So that's why it's super important to maintain homeostasis of their environment. Otherwise, they don't work well. Some of the key conditions we looked at over and over throughout the year are pH, temperature, and ionic conditions. So each enzyme works optimally at a specific range. And if you change that range, that can change the structure of the enzyme in such a way that it denatures them and they're no longer functional. They can't bind the substrate. Okay, that's molecular level. Let's move on to the cellular level. Okay, for the cellular level, we looked at the importance of cell size. Okay, in order to maintain homeostasis and balance and function and control, it's important that the cell remains small. Okay, the reason for this, remember, is that the smaller a cell, the larger its overall surface area to volume ratio, which is a good thing because the surface area provides for material surface, enough surface for things to travel, and the volume, you don't want it to be too big because then it's difficult to diffuse throughout the entire volume. Okay, so maintaining a large surface area to volume ratio is important. The other thing that's really important for homeostasis is you want to regulate what enters and leaves the cell. And that's done through the plasma membrane. So we looked at the structure of the plasma membrane, and we learned that it's semi-permeable or selectively permeable. And that means it only allows certain things to pass easily and others is more difficult, and they require the help of protein channels or transport proteins. So remember, the types of molecules that have an easy time passing through are those that are nonpolar, like steroids, or small, like water. Okay, so water is kind of the exciting. It's polar, but it's small enough to squeeze through the nonpolar tails. And then you have polar, ionic, and large molecules that have a hard time passing through. So they need help through facilitated diffusion or transport proteins. Okay, so surface area to volume ratio again needs to be large, and then we have the evolution of these larger eukaryotic cells. And just keep in mind the reason we can have these larger cells is because you now have these tinier cells or organelles that have membranes surrounding them. Okay, so you are able to compartmentalize specific functions by still maintaining large surface area to volume ratios through those membranes that surround each organelle. Okay, so compartmentalization is possible because you have membrane-bound organelles that keeps the surface area to volume ratio high. Okay, so a key aspect of the endomembrane system is the production and then the transport or secretion of proteins. Okay, so again, surface area to volume ratio is large because you have membrane-bound organelles that allow cells to now be bigger. So eukaryotic cells are bigger than prokaryotic cells. Let's focus on secretion of proteins. Remember, the step of making protein starts in the nucleus. And then from the nucleus, you move on to the ribosome that actually produces the proteins. Inside the nucleus is the DNA, which gets transcribed to mRNA, and then on the ribosome, it gets translated to protein. Okay, so DNA gets transcribed to mRNA and gets translated to protein. So we'll spend some time later reviewing some of that. Okay, so the ribosomes make the protein. Some of the ribosomes are actually bound on the surface of rough ERs, or rough endoplasmic reticulum. So once the protein is made, it enters the lumen of the rough endoplasmic reticulum. It's processed there, it's modified, and then it's surrounded by a membrane forming a vesicle and moves it to the Golgi. The Golgi continues to process the protein and finally packages it for delivery. So once it's packaged, the protein's found in a vesicle, and then its destination is either inside or outside of the cell. Okay, so I think that's it for cellular. The last level, and I'm out of space, so I'm gonna move to the left. Okay, is the organismal level. So on a larger scale, some of the things that we looked at in the organismal level is growth and development, and that's actually part of the big idea. So during the developmental stage of the organism, we looked at what determines how cells that become the organism, how those cells find their role, or how do they become determined and then differentiated. So remember, that's all about gene expression. All the cells in your body have the exact same DNA, and yet different cells have different functions. So how is that possible? It's possible because you have differential gene expression that turns on different transcription factors, and those transcription factors turn on tissue-specific genes. Tissue-specific genes. Cell differentiation is determined by gene expression, which then turns on transcription factors, and those transcription factors find promoters for specific genes. We also looked at induction very, very early on during development. You have a ball of cells, and some of those cells start to produce signals. Remember, there are certain cytoplasmic determinants that turn on those signals, and those signals diffuse to neighbors and influence the activation of specific transcription factors in those cells. Okay, and then finally, we also looked at apoptosis and the role of apoptosis in development and growth. The most memorable example is probably the shaping of your digits. So initially you have these webbed fingers, and then through apoptosis, the webs, those cells die off, giving you your digits and your fingers and your toes. Okay, so I was able to squeeze everything in here. Let me color code the different topics that we went over. So for energy, I'm going to use blue. So we talked about how biological systems utilize free energy, and the two processes, photosynthesis and cellular respiration, are really important for that. And then we looked at some biochemistry and molecular building blocks, how monomers are joined to form polymers. I'm using orange for that. So put a box around dehydration synthesis and our organic compounds and how they're formed. And then homeostasis was a big topic, so I'll use red. And that's pretty much the rest of the page. Homeostasis, how do we maintain constant conditions? So this is a nice way of looking at the big picture. It's your job now to fill in the details and continue the review by looking at the framework for AP Bio, but I hope this will help you kind of make sense of this big, big, broad topic.